certain cohort, and currently they reported that there is no currently CDC report. It says that there is no approved dengue vaccine. At the same time, we know that thousands of people are affected by dengue every year. So we had today, we have Takeda vaccine. It's a 2-injection vaccine registered on April 10th, 2025, and has been certified by WHO. The efficacy against hospitalization is 80% after 54 months.
Butantan in Brazil requires only 1 injection, which is very good, and it is going through the 3rd phase of clinical trials, and 60–70% of efficiency without hospitalization. Both vaccines are quite safe, and we don't have ADE there. And what is planned for tomorrow in terms of dengue vaccine? A recombinant vaccine that is based on fusion surface protein that includes all 4 serotypes. 1, 2, 3. Here it is. Sorry, this is the slide that I have already mentioned. Here we are. So this is the vaccine that gives VLPs and Innovias with the DNA, with the fusion proteins, and our products, experimental ones that we are working on in the Federal Medical and Biological Agency, and these are recombinant proteins, also surface ones, and the capsid ones that we have already used before. The origins of the new concepts are in ConvaCell, as I have previously mentioned, and there was an article published in 2019 that mentioned that dengue, as well as ConvaCell and coronavirus, can be used with the capsid protein, as it was told in this article. We can see it here, just a second. Here it is. And it was mentioned that this internal capsid protein will be the main contester for the vaccine. And how does the nucleocapsid protein work? It is expressed in large quantities. How is the vaccine different from the infection? And it can cause and can lead to immunosuppression, and why a person does not develop immunity and can get sick one more time, and it is because it blocks the immune response, and it can be eliminated because there is no memory cell. And how does it work with the infection? It appears there on the surface of infected cells, and with the help of this response, it stops the propagation of the disease. And what is the difference between vaccine and infection? There are many publications dedicated to this topic, and all of them demonstrate what happens with the infection, with the immunosuppression, but with vaccination we don't have such a reaction. So here is the strategy of the virus propagation. It shows that nucleocapsid proteins are the main ones in immunosuppression. So what happens when we use nucleocapsid protein in vaccination? It is injected in a healthy person, and it has a normal response, and the quantity of the protein is much less and can't cause immunosuppression. And the formulation of vaccine uses the special blocking VLPs and excipients that improve and increase a normal immune response. And on the way towards new vaccines, we are not only talking about nucleocapsid protein for normal viruses, but also there are a lot of cross-sections in those types of the proteins, and it can have a uniform, a universal solution for Zika virus, for example. Of course, it is another family of viruses, but still, for the Western African disease, it can prove to be efficient as well. And the experiments that we have done have shown that for cross-neutralization, in some cases, we don't see the reaction, but in particular, for some live vaccine, yellow fever and Japanese encephalitis, we have seen this type of cross effects in yellow fever and in Japanese encephalitis. The protection was demonstrated and was proven both in animals and in people. What is the potential for this concept, not only for the dengue, but also for other viruses? And immunologically, they show the same results, and we can actually hope that other virus infections can be tackled with such an approach and such a concept. With that, I would like to conclude my report. Thank you very much for your attention. And now we will go to the next report, and meanwhile we will be gathering the questions, as I've previously told you. Now, I would like to give the floor to Mr. Sebastian Rabdano, and he will be talking about the new strategy and the platform innovative solutions in the development of immunobiological pharmaceuticals. Please, the floor is yours. No. The replacement, Ms. Yelena Bogoslovskaya, she will be joining us online, and then we will listen to Mr. Rabdano. That is how we decided to do that. Good morning or good evening, Yelena. I will give you the floor now. Can you hear us? Can you see us?
Can you hear us and can you see us?
Dear colleagues, I think we have some problems with the sound.
We are having problems with the sound at the moment.
Dear colleagues, can we resolve this issue with the technical problems? Then we can go back to Ms. Bogoslovskaya later, and we will proceed with another report. So I suggest we should start again with Mr. Rabdano. Dear colleagues, good morning, everyone. And I'm very glad to present to you our developments and our ideas at this remarkable Congress between Russia and Nicaragua. And I think that today I will be focusing on how platform solutions help us to implement and introduce innovations very fast, as it is necessary for people, for our consumers that need vaccines and new pharmaceuticals. So, I will be talking about which platform solutions we have and which innovations can be used with the help of those solutions. And I'd like to start with what challenges we are facing in pharmaceutical industry. What does the society expect from the developers of those pharmaceuticals? They have to be safe, they have to be efficient, and they have to help people. And of course, they have to have affordable prices. And as we don't have the necessary medicine for all illnesses and for critical diseases, we need for the development to be very fast, and that is the challenge we are facing. We need large-scale production in order to provide an access for all those who need this type of pharmaceuticals and vaccines, especially for the limited series. And of course, all the pharmaceuticals should have a high quality so that the consumers should have trust in them. Of course, so all of us want to go through the registration procedures as soon as possible, as fast as possible. We all want to use the most state-of-the-art advanced technologies that will allow us to provide for high efficiency without ADE. And these are the requests, these are the needs that we see as the developers of pharmaceuticals, and we need to satisfy all these demands in order to be successful for the benefit of all the people. And this is a very good approach. We can cover all those needs with the platform approach when we use some validated blocks that have been tested many times. We feel quite confident in them, and we don't need to start from scratch with the scientific base, all the necessary proofs. And such an approach allows us to cover all those and satisfy all those needs and to have a response to all these challenges. We have three platform solutions at the Thrives Institute, and this serves as a basis for us. The virus platform, on the basis of chick embryos, we produce influenza vaccines that have proven its efficiency for a very long time already. Another important platform is for Haemophilus influenza, the recombinant basis, and virtually we can create actually any pathogen, any antigen that can be reproduced with some restraints and some limits with the protein expression. On this platform, we have created a Convacell vaccine that we all know quite well. And a new platform also includes polysaccharide antigen reception, the conjugated vaccines with proteins in order to improve the immune response and to launch the necessary mechanisms in immunity. And we are creating, developing and producing new pharmaceuticals, and we have had some successful results that enable us to have a smooth transition from one point to another. The 5 blocks of the technologies, they can be substituted and replaced, and I will tell you more about the differences between the blocks, but they can be reproduced, they can be used in all the stages of the production cycle, and it helps us to shorten the time that is required for that. In particular, with virus platform, at the beginning, we get the strain of virus from isolates from their collections, and then we prepare the banks, the working banks, and also the lines of the chick embryos. Then we infect and inoculate them, and then the antigen is used as a substance for the production of vaccines from the same blocks on the same virus platform. We can create other pharmaceuticals, not only influenza vaccines, but we don't need to create each block from scratch, and it gives us a lot of advantages. As for recombinant platform, the approach is quite similar, but this is a synthetic type of strain, the genetic code for the protein that is introduced into the strain, and we start with the same scheme of the banks, and we start to produce the vaccine.
As for bacterial platform, everything starts with the isolates of strains, characterization of those strains, creation of culture banks, purification, isolation of polysaccharides, and synthesis of the same combinant platform, the proteins, which are subsequently conjugated and purified, and obtaining the substance for the vaccine. These are the main blocks used to build any of our developments. And on these platforms, we are able to create both viral, bacterial, and therapeutical medications as well. When I said that we have a lot of achievements, please look at the time scale. Here we see that for each of these platforms we have 1, 2, or 3 medications, and for the future we have plans for a range of medications which we will develop and launch into the market. Among them, I would like to specifically mention the dengue vaccine, because at this development we have been focused recently and intensely, and we have obtained the first results, which I will present in great detail. I will have a couple of slides dedicated to dengue vaccine, but in order for general understanding how we expand our portfolio and how do we create the new vaccines, either way, in each of 3 platforms, we have this process of creation of this strain, producing strain, be it viral or bacterial strain. These are the producents which are programmed. These are programmed biological tools to create the molecules we need, which we use as a substance for our medications and for the vaccines. And from this producent, we obtain a large amount of biomass, isolate, purify the proteins that we need. This approach was used in the development of flu vaccines, H5N1 bird flu, human papillomavirus vaccine, our own development. We have diagnostic medications for TB and some other medications which I will not dwell in detail at. So in order to demonstrate how the platform approach assists, I will use the example of the dengue vaccine, which is now in active development stage. We are approaching to the selection of antigen in the systemic way. This is the stage of the choice of antigen, which is different for different medications. We are unable, we have to, this stage is unique for each medication. We cannot use generalizations here, since medications have their own genes for their proteins. This stage will also be unique for each of the medications. And then we are using blocks and developments which we kept after COVID vaccine development. The parent strain we already have in all the laboratories. They are characterized. We have well trusted and trained procedures to create the bank and technological process for the cultivation for recombinant strains. We do have the most efficient pathways with high yields, which decreases the cost of the vaccine to acceptable levels, which influences accessibility. The purification methods for other recombinant proteins can be also transferred for other medications. So if you look at the number of stages which are unique for the medication and compare to those who are may be substituted, and the majority of stages may be reused and may be used again and again after other developments, which allows us to create the required results, giving us advantage in time and cost. We do not need to create all the new documentation since the majority of documents are already available, just needing minor amendments which are unique for each medication. So based on the example of the dengue vaccine, we can demonstrate it very well. The platform approach stages allow us to save time and money. This is one of the most interesting slides in my presentation. In dengue vaccine development, we are working with three versions of vaccine. One is based on E protein, and two are based C protein based. In 2025, this year, we are holding preclinical trials, preclinical studies, in order to estimate the efficacy on animals, and then we move to empirical studies. For this medication, we already have capacities for its production, industrial capacity. As soon as its efficacy and safety will be fully confirmed, we can immediately transfer to the mass production, producing millions of doses within few months, since we already have everything for recombinant platform. We have experts, we have raw materials, we have equipment, we have methods of quality control. So these are the advantages which are extremely important here. Here at this slide on the right, you see preliminary results of in vitro studies of one of the versions of the vaccine, where we have shown the titer neutralization, and neutralization titers are over 2,000. Therefore, we are sure that at least one of these versions will be highly efficient. However, we also study all the versions for their efficacy and safety. So once we collect all the information, we'll publish it in scientific magazines. Our strategy for this vaccine is as follows. Dengue is not endemic infection for Russia, but it is endemic for the Central America and Latin America. Therefore, our strategy is defined as follows. The development and research is both in Russia and in Nicaragua. The production of the substance will be in Russia, since based on the platform approach, we have all the necessary equipment and capacities. So but the production of the dosages of the final medication is quite logical to base here to provide access to all those in need. And clinical studies of efficacy are impossible in Russia because only the risk groups may be immunized with this vaccine and monitored. So it's definitely pays to have it here, and the application of vaccine in its endemic region for the disease is the basis of this strategy. In general, I would like to say that besides platform approach, which already include innovations and high technologies, in the Russian Federation we have a number of other developments, and the Federal Medical Biological Agency has includes a number of institutions which are at the front line, and their developments should be applied into practice, such as vector mRNA vaccines, cell products. All these is implemented in Russia through our institutions, and these technologies, these approaches may be exported, as well as the know-how and expertise may be transferred using the available institutions. It's also a platform approach to the transfer of technologies and sharing experience. The platform solutions of SRIPS have proven their efficacy on the example of the medications I have demonstrated. So now it's evident the advantage of platform approach is evident now, which is in the basis of unification of manufacturing production process, analytical methods, which also decreases the launch to the market time. For example, for Convacell vaccine, from the beginning of its development to the first commercial series it took less than 2 years, which probably is quite in contrast from the textbook, which claim that a new medication requires 10 to 15 years. No, it's wrong. If you use the platform approach, if the pathway mechanism has been already tested, this is the new reality in which we live. For us, the international cooperation with Nicaragua is very important. It's strategically important because it allows us to view in a new light the infections which exist in globally, and opens our opportunities to export our know-how, technologies, and experience, thus sharing our developments. Thank you. Dear colleagues, we have listened to a very important presentation. Therefore, we are behind schedule. So please stick to your time slot. Dear colleagues, what about Moscow? Is Moscow online? Can we hear the presentation from Moscow? If everything's okay, please turn on. I see that not everything is okay so far. While we are into technical issues and they're being resolved, please think of the questions. You see there is a QR code on the panel which will help you to go online with the questions you might want to ask. Probably at the end we'll be able to provide brief answers to your questions. Would you, Georgy Mikhailovich, would you be able to present this topic since you are part of the developers team? Dear colleagues, we have an expert who is ready to comment on the presentation we have not heard. Georgy Ignatiev is the expert in virusology, including dengue. Yesterday, there was his presentation on chikungunya. So his opinion is very important to us, both from the point of view of the strategy development and tactics. And I would like to invite him to comment on the presentation we have just heard, and also to briefly comment the presentation we were unable to hear online, since he is a co-presenter, so he is entitled to to this presentation.
Thank you. Dear colleagues, yes, I know the contents of Ms. Bogoslovskaya's work, and maybe she will join us later, but making conclusions about the previous two reports and the report by Ms. Bogoslovskaya, I can tell you the following. Of course, our agency is working on the development of the vaccine on both platforms. The one in Thrives, and another one is the Center for Strategic Planning using the recombinant strain. And this strain includes three determinants and of all the four serotypes. As of today, colleagues have studied immunogenicity of this vaccine, the formation of antibodies, the reaction of neutralization and immunofermentation, and they have estimated and evaluated the effect on the serums after the immunization of mice. And the data that I have at my disposal at certain points, these serums don't provoke any ADEs or replication of a virus. The next slide. No, this is not the one we need. And getting back to the developments of our strategic center, they evaluated immunogenicity and the absence of ADE effect, and they carried out the research that allows us to say that this vaccine is quite safe on this scale that is stipulated by the Russian legislation, the toxicity, the formation of the necessary cellular immunity and the blood elements reaction and its effect on the central nervous system. And that is the level of the development at our Center for Strategic Planning. And our colleagues, we see with our colleagues that it is not only about the method of control, but also the products that will help us to evaluate immunogenicity of this pharmaceutical in future. And they provided a set for antibodies for dengue virus, which is quite specific cross-reactivity with the other viruses of the same nature. Encephalitis that is not endemic for Central America, also Western Nile and yellow fever that said does not provide the false positive, but it also provides the serums from people who already suffered, who already had dengue, and also the data that we can use from the experiments with the mice. And it does not provoke any cross reaction with the Western Nile fever. And that accompanying product does exist. It allows us to perform the CAP screening at the preclinical stage. There we evaluate the effect at this stage. And I think that this type of support should be worked on also for the second platform, the recombinant proteins, or maybe we should combine those two products. But taking into consideration the reports that we've listened to and the report that we have not listened to so far, the approach is not only about the development of technology. efficient technology, but it is also about the safety of this product, of this pharmaceutical that we will get as the result of the application of this technology. And it applies to all the products that are developed at St. Petersburg Institute and at the Center for Strategic Planning. And it is about their safety. We should focus on that matter, and the absence of antibodies should it be considered as a heal-all, and the absence of ADEs. We should focus more on their safety issues and all those, or on all those stages, and then we can start with the clinical trials. But as I see it, dengue fever as a disease is not endemic for Russia, and our colleagues from Nicaragua have much more extensive experience of working with dengue as a virus and serum from those who have already had this disease. And probably we should continue with a collaboration with our institutes in Russia, with our Nicaraguan colleagues at the stage of the preclinical trials, the research protocol preparation. We do need this cooperation right now. All those developments should be estimated based on the immunopathogenesis of this disease, and our colleagues, I think, have much more experience in this area. And that is all I wanted to say. Thank you. Thank you very much, Georgy Mikhailovich. And I think that your opinion is quite valuable and it can help all the developers. But I can argue with you in one thing. I think intrinsic proteins are not seen as potential provokers of ADs. They are not on the surface. That's why the mechanism is different. We can hypothetically think that it exists, but we haven't seen that in practice. And I have a question to our technical team. Moscow has joined us or not? Yes, seems that it works. And then the next report will be delivered by Mr. Sidorenko, the specialist in bacterial infections from our institute, from the Federal Microbiological Agency. And I would like to give the floor to Mr. Sidorenko. Is he joining us? Can you hear me? Sergey, can you hear me? Once again, good morning, dear colleagues. Thank you for the invitation to speak at this Congress. And the previous reports addressed antivirus vaccines, and I'd like to talk more about bacterial vaccines. But due to the biological structure, the main antigen for almost all bacteria is the surface polysaccharides. But also due to the peculiarities and characteristic of immune response, the pure polysaccharide vaccines are not sufficient in terms of immunogenicity, and this is an obstacle, and we resolve that by conjugation with some carrier proteins, and we have had some experience with these carrier proteins. We've demonstrated its efficiency, but also the bacterial pathogens. As in many other cases, we see serious problems. The changes of the external structures of polysaccharides. One of the most problematic issues that we see in pathogens is the pneumococcus. And A lot of publications have been dedicated to that. Here we see more than 100 polysaccharides, and to prevent such types of infections, we should develop 100 vaccines against all those pathogens. Technologically speaking, it is quite complicated. In the foreseeable future, we will not be having opportunities to come up with a uniform protein vaccine. So the fact that the SRIFs and the Matchnikov Institute in Nicaragua, they have developed a ready-made platform to structure a polysaccharide vaccine, and it is great, and it has good prospects. But a huge problem is not only the changes in pneumococcus, but dynamic changes. And we see that in 7-8 years after the first use of pneumococcus vaccines with the polysaccharide formulation, we started with 7 valent, 10, then 12 valent and vaccines. And now the market, we have 15, 20 valent vaccines, and we can have even more than 30 valents. That was what we are going towards. And in order to battle efficiently against those infections, we should organize efficient monitoring and observation of the serotypes that are included there in certain regions. And we are facing the following problem. As of today, all the vaccines that have been developed by our Western colleagues in terms of the serotypes do not satisfy our needs, our local needs. And in substance, we should follow the way with the platform solutions in order to do that quite fast and change the polysaccharide formulation of the vaccine, the same thing that happens with the influenza vaccines. And it is a really huge problem, first of all in terms of the regulation, but we have to find a solution. We have to resolve that, because now when we work on a uniform conjugated vaccine for all geographic regions, it seems quite impossible. Another problem that we are facing is a technological one. As of today, none of the pneumococcus vaccines has not shown efficient results against pneumococcus. Of the third serotypes, we've applied a lot of the vaccines. The share of the third serotype remains the same, but it is the one that leads to the most severe cases, with the death rate that is quite high. And the conjugation of the third serotype of the polysaccharide vaccine is very problematic in terms of finding an optimal solution. And of course, we would like our colleagues, the developers, to pay attention to that process. Meningococcal vaccines, it's a little bit less complicated. The number of the polysaccharides is more limited, is much less, but another problem arises there. The protection against meningococcus of B type for the polysaccharide are not efficient against that, and there is a hope that the groundwork that has been done at SRIFs will help us to overcome this obstacle. And I would like also to mention that the polysaccharide conjugated vaccine platform opens good prospects to fight against not only traditional diseases, meningococcal, pneumococcal infections, but also the other ones. We have to fight against the pathogens of Sapsiella, Escherichia, and other types like Staphylococcus. And of course, we would like our colleagues, the developers, to think about these problems, to take them into consideration, because these are very severe problems for the healthcare. Thank you very much for your attention. Thank you, Sergey. And I'd like to ask a question one more time. Is Ms. Bogoslovskaya joining us? Then we go back to the previous report that we have not been able to listen to. But we are quite flexible in this respect. Dear colleagues, can you hear me, dear colleagues? Can you hear me? Can I start? Excellent. Dear colleagues, I will present the results of our dengue vaccine for the prevention that we have developed at our strategic center. Unfortunately, I don't see my slide, and I will not go deep into the detail because Sergey has already told you about the registered vaccines. I'd like to repeat what has been said about the relevance of the efficient vaccines.
The next slide, please. During the development of our proprietary vaccine, we have paid special attention to the antigen selection. It was demonstrated that immune response for different structures is different. So to this surface proteins cause the creation of antibodies. On the other hand, it was been demonstrated that they were specific with a low ADE potential. Next slide, please. And the equalization of amino acid sequence of serotypes showed the high efficacy of this protein. We have constructed the sequence of antigen, which includes the D3 domain with 4 serotypes conjugated. The obtained antigen, called D34, has been built into the vector, and this development has been patented in Russia. The next slide, please. I have to mention that we have selected D-cell epitopes from conservative dengue protein, which have combined in one sequence and introduced them into the vector, which has to provide cell immune response and increase the safety of the vaccine. Next slide, please. To estimate the immunogenicity of the vaccine, we have jointly worked with the Institute of Vaccines in Moscow to develop the reagents to identify antibodies for D34. It has been tested at patients' serums compared to a bot kit for dengue, and the concordance was over 99%. The vaccine we have developed, called DengueVac, has been studied for immunogenic properties. The results have been published in 2023 and has been demonstrated that when introduction of the cells of this strain, there is a high expression of antigen D34. The mice have been twice immunized with the vaccine in 3 weeks, and in 3 weeks after the immunization, they have demonstrated antibodies in high titer. The antibodies developed were equally efficient with the brain extract of 4 serotypes of dengue virus, as well as have shown neutralizing activity against the serotype 2 of the virus. In the last 6 months, we have made broader studies, mainly studies of the antibodies titer after triple vaccination. The highest titer is in 3 weeks after immunization, then it goes down and is kept for the period of observation as 1 to 1600. Similar neutralizing activity has been demonstrated against all the four serotypes of dengue virus. And finally, we have observed that the titer of neutralizing antibodies average was 1 to 900, 1 to 800. Jointly with the Research Institute with Mechnikov Institute in Moscow, we have estimated AD effect in vitro. This model included parallel estimate of Vero cells neutralization and AD effect on K562 cells, which are poorly infected with the virus, but due to the expression of the virus, they provide the entrance to the cell. So we have used the immunized mice cells, and the positive controls we have obtained, positive standards we have obtained mice after infection with one of four serotypes of virus D. It's called D1, D3, D2, D3, and D4. The results show that the high neutralizing efficacy, high neutralizing activity, are the serums obtained after dengue-vac immunization, and in control groups from mono serum D1 and D3, K562 cells. The increase of activity was used after the usage of the serums D1, D2, D3. When using the dengue-vac serum, none of the viruses have demonstrated the increase of infection of efficacy. And now we are developing the vaccine for mice model for lethal and sublethal dengue. Know that the regular mice are not susceptible to dengue virus, are not infected, but we have developed a model with gene interferon receptors. Mice G129 have interferon of type 2. Both lines of mice are infected with dengue virus without lethal outcome, but the usage of two genes may provide lethality. We are adapting now dengue virus to the mice of A129 using mice with two knockouts and using the line of A129 to estimate ADE. Prior to the end of the year, this research will be finished. And finally, we have developed a candidate dengue-vac vaccine, which has high immunogenicity, providing the formation of antibodies neutralizing four serotypes of dengue virus. The candidate vaccine dengue-vac has demonstrated the absence of AD effect in vitro. Prior to the end of this year, we'll finish the studies of protective activity and clinical safety studies, and will prepare the documentation to obtain research permit for clinical trials in Russia. Thank you for your attention. Thank you, Yelena. I apologize for the absence of the slides here. The presentation has not been loaded for some reason. It happens. Nevertheless, we proceed with our session. The commentaries of Professor Sindarenko of pneumocoque and meningocoque vaccines have been very important. We are working closely with this person and with this institute for the development. So next to me I have Mr. Belankin, the head of the developers, the head of the department for the development of bacterial vaccines. So the floor is yours, Mr. Belankin. Good afternoon. First of all, thank you very much for the opportunity to speak here. I would like to add the presentation of Sergei Vladimirovich on pneumocoque infections. He has rightly observed that we know a number of serotypes of pneumocoque, over 100 serotypes. Is the answer to create a 100-valent vaccine? And we think that no. And the development of centavalent vaccine would take years and years and decades. And what if serotype 101, 102 serotype will appear, which might be even more dangerous? So among the number of pneumococcus serotypes, this disease is well known. Only 20 to 30 are causing the disease, so we have to target those serotypes. These occurrence of these serotypes highly depends on geographical region. Currently, pneumococcus are tridecavalent. What the consequence of the application? The tridecavalent vaccine has been efficient against serotypes which are included into it. However, its application resulted in the proliferation of serotypes which were not covered by the vaccine. Therefore, it could lose its efficacy with time. Therefore, we have to increase serotype composition or probably renew it. What do we need to create new serotype composition? We need cooperation of the diagnostics, physicians, infectionists, and manufacturers and developers of the vaccine. This is the way to create a maximum efficacy vaccine against pneumococcus. Thank you. Thank you, Andrei. I would like to add and comment. Pentadecavalent vaccine is one vaccine. Pentadecavalent, well, it means it has 15 vaccines. We already have said 16 vaccines. It's not something with 15 buttons on it. No, we have developed 16 vaccines. Therefore, I'd like to draw your attention to the fact that both scientific potential and economical potential do not allow to develop all the vaccines. So we call for the development of universal vaccines. And based on the example of pneumococcus vaccine, which has tens of serotypes, we have to develop some kind of universal vaccine. Thank you for your attention. Each medication is registered after its development, and here we encounter a number of complications. Since the scientists are not quite familiar with regulatory process, we are dealing with mechanisms. Once it comes to registration, there are a number of details. And Yelena, I think next to me, Yelena Rozanova, is an expert in registration who assists us, the scientists, to overcome these issues in advance. And I would like her to present on the peculiar features of the final stages of development of clinical trials. Everything that awaits us during the registration, what are the issues there? The regulatory activity is improved, it's modified, it's changed. Things we have done yesterday, we have to do differently today, because today they are already part of the new pharmacopoeia rules. This floor is yours. Good afternoon. Thank you, Sergey, for the opportunity to speak here. I would like to continue and to enhance the discussion about recombinant platform and polysaccharide platform, since we are talking about platform approach. And first of all, I would like to dwell on recombinant platform and say a few words about Convasil vaccine, which has facilitated this conversation that we are actively discussing dengue vaccine and are able to develop it. Convasil vaccine, we have presented several times during the Congress. We present our achievements, but despite the fact that last year we have received the registration certificate in the territory of the Russian Federation, we did not stop on that, but we are now running clinical trials of this vaccine in the territory of the Republic of Nicaragua. These clinical trials are using 3 dosages, ampoules, produced by our institute, SRIFs, 1 dosage vial and 6 dosage vial, and 6 vial 6R, which have been produced in Nicaragua. Therefore, in order to use this form in clinical trials, we have done both clinical and analytical studies. We have been inspected, all our manufacturing sites have been inspected by SecMed, and we have run a huge volume of work alongside with technology transfer and other aspects. We, jointly with Mechnikov team, SRIFs, people who are involved in clinical studies, and Mechnikov team, we have jointly prepared ourselves for clinical studies, developing the protocol which includes international and local regulation. And we have studied a number of cases which, for the dossier, which we had to overcome. Currently, clinical trials are on the way. They are in active phase. 300 people are vaccinated already, and we plan to recruit the entire group by September. And day 42 after the immunization, we'll estimate the immunogenicity in our branch of SRIFs in the territory of Nicaragua. As for registration activity, as I have mentioned, in the Russian Federation, we do have permanent registration, and in the Republic of Nicaragua, we are planning soon to obtain the registration for both dosages, 1 and multi-dosage vials, since the dossier has been already presented to the Ministry of Health of Nicaragua.
Also, I'd like to talk about the processes and our success and achievements in the development of polysaccharide vaccines. What has been mentioned by Sebastian in his speech, the first vaccine, baby hip, that has been used for the prevention of a Haemophilus infection. It has been registered on the territory of the Russian Federation in April 2024, and in 1 year, more than 300,000 dosages have been used on the territory of the Russian Federation. Why have we been able to do that so fast? Thanks to the platform solutions, approaches, and also the interaction between registration and the transfer teams and research and production. We have taken into consideration our previous experience with other vaccines, and that is why we started the commercial sales so fast. Now we are getting ready for the registration process on the territory of the Nicaraguan Republic, and the dossier is being translated, and we will submit it quite soon. As for the pneumococcal vaccine for the prevention, it's a huge discussion and debate how many serotypes we should include in the vaccine. And now we have 16 serotypes that are widespread on both Russia and Nicaragua. Our colleagues have conducted an additional epidemiological analysis about the propagation, and we've received the final report for the second and third stage, where we evaluated the safety, and it is even safer than the Prevenor, the standard sample we compared it with, but also immunogenicity. And in order for us to be quite confident about efficiency of this vaccine, we studied at this stage the functional antibodies production, and for the serotypes that coincide in our vaccines, the production of antibodies is the same. And as for the unique serotypes, the 12, 13, and 16F, against the backdrop of our vaccine, the level of antibodies is higher than in the standard sample. And now we are completing the third stage of clinical trials. And we are getting ready to do the research at the children's population, because it is used primarily for children and only then for adults and elderly population. At the end of this year, we will submit the registration dossier in the Russian Federation, and taking into consideration a very short timeline and our ambitious plans, we are in close interaction and cooperation with each other, and we share all the news in a timely manner. And if some problems arise, we resolve them jointly. And at the end of my presentation, I'd like to talk about meningococcal infection vaccine, and it combines polysaccharide platform and recombinant platform, which is quite interesting. And the developers, the guys that are working on that, they have made a discovery, I think, that I think will help to safeguard and to protect the population of the world against some infections. And we are completing the first phase, and we already know that this vaccine is quite safe, and we are getting ready for the clinical trials with the participation of both adults and children. Also, we'll start the registration process, and in future, I hope that these vaccines will be produced both by the capacities of Mechnikov Institute and SRIFS, and the Republic of Nicaragua will have an access to these vaccines. Thank you, Elena. Dear colleagues, during all the reports, we have been talking about vaccines, and of course it is one of the main areas in terms of the prevention and the fight against diseases. But we are making progress in science, and we are studying new mechanisms, and we are coming up with new strategies. And of course, the tools of our weapons against infections will get broader with time, and it is not only about infections and about pharmaceuticals, also in oncology. And another speaker that we have, he's a director of the Center of Immunology from Federal Microbiological Agency. Can you hear me? Good morning, dear friends and colleagues. It is an honor for me to present at the hospitable land at Nicaragua, and it is not my first Congress. I'd like to say that I'm so glad to be here and to be able to share with you the results that we obtained in developing new pharmaceuticals. And we have mentioned quite a few times today about the prospects of the platform approach in many ways. And as for our institute of the Federal Microbiological Agency, I'd like to tell you more about that. The first platform is for the development of new dosages of RNA mechanism. At our institute, this platform is based on the scientific base polynucleotide synthesis, and it has been supported and funded by the government of the Russian Federation, and interference mechanism has been studied by us for more than 15 years. It exists in almost all the cells, and it blocks the expression and production of almost all the genes. And I can also say that it has been it was discovered in 1998 2008, and 6 years later, they were awarded a Nobel Prize. Why did it happen so fast? It became evident that this mechanism should be used for producing and manufacturing new pharmaceuticals, and that is what we did. For 15 years, we have been studying this mechanism to come up with new pharmaceuticals, and on the first stage, we developed a pharmaceutical against bronchial asthma, and more than 200 million, and this is a serious problem also in Latin American countries. And serotyping for the asthma showed us that interleukin 13 was the main one that leads to the so-called shift in the immune system to Th2 that is quite characteristic of the allergic reaction. And the first product that we developed on the basis of it is the one that blocks, and this is an inhalator, blocks the expression. It successfully passed all the clinical trials, and we published a very good article back in 2014 about the clinical trials. On the next stage, we upgraded this product for the blockade of the expression of the interleukin 13. It is also an inhalator for the therapy not only of asthma but also allergic rhinitis. And in 2019 we should have started clinical trials of this pharmaceutical, but then came 2020, and the world has completely changed because of the coronavirus pandemic. And Ms. Krasova, the head of our agency, has set a task for us, an antivirus product against coronavirus infection that would be based on the same mechanism, and we successfully did that together with the colleagues from St. Petersburg Institute, together with Alina, Sebastian, Sergey. In 2 years, we went a long way, and we launched this product on the market. It successfully passed all the stages of preclinical and clinical trials. In 2022, we launched that on the market. It has a permanent registration certificate right now, and all the stages of the research, the preclinical trials, clinical trials, the first, second stage, and the third stage have been successfully completed. We've demonstrated the efficiency, the safety of this product. It is very efficient in terms of the therapy against the coronavirus infection, and the results were published in the Allergy Journal, one of the most famous ones in the world. And you can actually look up those results. They are in open access. And we created the whole platform that helps us to develop many products, many pharmaceuticals for bronchial asthma, and also another products against the ischemic disease of the heart, and the small interfering RNAs and atherosclerosis prevention, and the ischemic heart disease. We are also developing pharmaceuticals on inflammatory diseases and neurodegenerative diseases. So this is the first platform I wanted to mention, and it helps us to develop new pharmaceuticals on the basis of the interference mechanism. The second platform is used for recombinant allergic vaccines. It is quite well known, in spite of the fact that the pandemic of coronavirus has come to an end. In terms of the allergic diseases, they are still here, and the reasons for those diseases and their severity will be growing, and to other factors like global warming and others. And we have more than 1 billion patients that are suffering from those symptoms. So fighting against allergy is a very relevant challenge. And we developed recombinant vaccine for the allergic reactions. In Russia, we have an acute problem with the reaction to birch, and the same applies to Central Europe, Japan, Australia. More than 100 million people are suffering from this allergic reaction to the birch, and it propagates on very large territories, and it causes a very high degree of inflammation in the noses. So our task was to develop a recombinant vaccine for that. We have an international team together with Professor Valento from the university. We worked on that, and we can say that we did develop this first of its type recombinant vaccine. It is safe, and it includes the immunodominant elements of the birch and apple. And when we introduce that, it produces the necessary antigens that are blocking an allergic reaction. We successfully completed clinical trials of this vaccine. We demonstrated its safety and its efficiency. In 5 injections, it demonstrated a good level of the production of LPG bodies, and it also has an inhibition reaction, and it helped to block the allergic reaction in patients.
There is also food allergy associated with the birch tree, and we have demonstrated that this vaccine in preclinical trials have protected from the associated food allergy like apple, nuts, soya, and peach. The clinical studies of this vaccine have been started last year. Currently, we are joined first phase 1 and phase 2. Phase 1 is completed successfully. Phase 2 now is running in Russia, since the pollination season of birch is now in Russia. So probably next time we'll be able to present the data for this phase 2. Not one vaccine has been developed. They have developed the platform to create recombinant vaccines jointly with our colleagues from Mechnikov Institute. We have discussed it in Managua, and we know quite well that in the countries of Latin America, there is a widespread allergy to seafood, fish, eggs, milk. So this platform allows us to create vaccines to fight the allergies I have mentioned. This is what we are currently doing. So once again, it's the platform approach is highly prospective to the creation of the new drugs, and we are successfully involved in it. Thank you for your attention. Thank you. Thank you, Gusar. I have mentioned the universal applicability of vaccines, so not just the number of allergenic agents development is very important. Who could have ever thought that the same vaccine will be able to work against allergy against birch pollen and apple tree? What's common between these two? Sometimes you cannot even come the joint features, but sometimes you should search for it and find it. And next presentation is by our colleague from Cuba, SecMed, Lizette Perez Ojeda, SecMed Advisory. These are people who are helping us to launch our medications and bring them to the end consumer.
the advances in the technological platforms. In the past few years, we have seen a profound change in the new platforms that permit more efficient development of vaccines. And these platforms, we need to ensure the quality of the treatment, and taking all this into account, see if the products reach our populations in a accessible and timely fashion. Thus, the innovative approaches on the part of regulating agencies, we have worked toward a proactive approach to regulation based on risk analysis and the cycle's life cycle and the technical accompaniment of the research phase. This, in the case of COVID, we have worked with technical accompaniment of the technology transfers that began as a experience with Menchikov and then also with Brazil. And here in Nicaragua, of course, is a very good example of regulating a company to reach the stage of approval. And we have all heard that one of the biggest successes has been that the vaccines have been pre-qualified by the World Health Organization, or uh, vaccines, COVID vaccines in Cuba, the Soberana, show that it is possible to have very quick movement in regulating adapting by adapting international standards to local situations. Um, and this has to do with the academic and scientific communities working closely with the state agencies. Also, the role of international cooperation, above all in regions such as ours in Latin America and the Caribbean, where we have to work on the strengthening of biological and harmonized criteria to be able to ensure the timely access that we are referring to here. And what is now being talked about a lot is a reliance or trust among the different regulatory agencies, and we are working to promote this type of actions, this type of reliance, to accelerate decision making as well as risk assessments between regulating agencies. And it's also important to mention that this is a work that we promote, but that is also very interesting, and we're working closely with our colleagues in Russia, some of which are present here. And to be innovative also means you need to look inward and incorporate digital tools to the process of regulations and prepare ourselves to evaluate complex products such as cellular therapy, genetic therapies, and vaccines such as those, the pentavalent that we have heard of, and that we have heard about lately. And the innovation, technological innovation, needs to be accompanied by ethical and responsible regulating. And so we are hoping for regulations that not just control, but also accompany and permit putting technology to the at the disposal of our peoples. Thank you very much.
Thank you very much, Lisette. And now the floor goes to Angela Garcia, National Regulatory Agency of Nicaragua. It is my special pleasure to do so because in the recent past, Nicaragua did not have its own regulatory agency. So we see not just the development of technologies, but we also see the development of regulatory activities, which is quite necessary for each country, otherwise unable to perform its activity in the healthcare area. So, Angel Garcia, NRO Nicaragua, the floor is yours.
Good morning. Thank you for the opportunity of participating in this Congress. Modern pharmaceutical industry has become a new and modern tool, especially for the development of vaccines, as my colleagues have said this morning. In Nicaragua, the strengthening of national capacities in this area is a great opportunity to improve public health for the scientific and technical development and the sanitary or health safety. Regulatory authorities need to be able to accompany this development with technical capacity, with an effective surveillance that guarantees that all products that are produced locally comply with the standards of safety and quality. And the authorities need to foster development but keep their authorities as well. The country is thus in the capacity of producing vaccines on the local market without depending on foreign markets. And this is important for health crises such as pandemics and others, where when the international availability of vaccines and medication might become difficult. The industry fosters not only employment but also public-private alliances. And there are many challenges for the regulatory authorities, such as the new technologies and the lack of precedence. Some platforms use innovative technologies, and the specific considerations are necessary for each product. Each platform has to prove itself to be adequate according to the product. We, as an authority, have to establish alliances with the academy, with scientific and research organizations, and we have to take opportunity of emerging technologies and foster public policies that foster national resources as strategic inputs. There has to be fluent communication between the different agents in this field, and the specific requirements have to be made known transparently and understood. In summary, the pharmaceutical technology is a specific tool for the development of public health in order to strengthen national sovereignty in this area, produce effective and safe medications and vaccines in accordance to the needs of the population. Thank you very much.
Dear colleagues, this brings our session to the end, and I have to say that for me, the ninth Congress, I have participated in of them, and we see changes for the better. We have new ideas, new medications, but one thing remains constant, our friendship and our cooperation. Let's keep it this way. Let's meet together, let's work together, and let's create new values. I have not seen if there were any questions submitted, but if there are, I would like to answer those questions.
is for Sebastian. Considering the difficulties in the development, in the previous development of vaccines, specifically what has been related with the effectiveness and risks associated independence of the antibodies, what do you consider has been different in the approach that has been displayed by the new candidates?
Thank you for your question.
Okay, eso funciona
I
mejor, ¿no?
believe
No,
that here, as Georgi have said, we see that potentially there are risks related to ADE effect. That's probably for the dengue the highest risk, and definitely we will include in the clinical trial list the estimate of this risk and its mitigation and minimization. But the most important thing is how we are trying to eliminate this risk, which immunological mechanisms we are implementing, which in our opinion will be able to avoid this risk. As I have demonstrated in my slides, we are currently working on 3 versions of the vaccine. One is based on E protein, which is surface protein, with which neutralizing antibodies causing ADE effect are associated. But here our approach is to create a high level of antibodies neutralizing in order to avoid circumvent this effect due to a strong immune response, which will not have neutralizing component which amplifies this effect. We are also studying 2 other virus protein. These are the same protein, 2 different vaccine forms. This is the internal protein, which is less related to ADE effect, because antibodies to this protein, they will not lead to the formation of immune complexes, which amplify the infection. So this is the fundamental approach, which we are currently trying to implement. But Dr. Rakelov has presented this concept in detail in his presentation.
The next question is about the it's very grateful for being here in front of the platform and the developers of this platform. This is a question that goes like this. In taking into account the products for infections caused by cell types that are not included in existing vaccines, could you please comment on whether the pentavalent vaccine that has been presented today is this or has found a solution of what strain to include in the case of the Russian Federation, or if you have any possibility to visualize this in relation to the new vaccines that have been appearing in different markets?
Thank you for your question. For hexadecavalent vaccine, we have used multi-factor approach to estimate of the serotype composition to make it most efficient, because the most important issue in the creation of the vaccine is probably the idea. That's the starting point. And from the right choice of this point, at this point, the entire life of the medication is depending. Has been proved by Conocell. Probably it will work well in our hexadeca pneumococ vaccine. We have estimated that the ability to cause the disease, the serotype, its current prevalence, based on the usage of heptavalent or tridecavalent. So we have estimated the prevalence of this or that serotype generally and locally in the territory of the Russian Federation. So due to this approach, which is now developing in our institute, likely will export this approach. So for each local outburst of pneumococ infection, we'll have serotype composition rather than creating 100-valent vaccine, 30-valent vaccine, probably for some regions, heptavalent or hexadecavalent composition, which will probably most universal for Russia and adjacent countries, but also probably will cover the need in pneumococ vaccines in the larger part of the world. Probably I have answered your questions. Thank you.
We have another question for Mr. Ravdanov. As to the plasmids that are using E. coli, how do they resolve the feeding for non-structural proteins that are important for the immunogenicity, and what is their limitation in this vehicle?
It's true that during the development of our version of recombinant vaccine against dengue fever, we have used bacterial platform, which has limitations on the reproduction of post-reproductive modifications, which sometimes are critically important in the formation of the right immune response with high specificity. Since it often happens that glycosylation causes very specific response to structural epitopes related to the glycosylation profile. We resolve this issue in the following manner. First, we use either epitope sites of proteins which do not carry glycoside, so we create highly specific immune response which does not depend on glycosylation by choice, by selection of epitopes. The other way is to use the proteins where we target D-cell immunity, where there is no need to create cell epitopes, structural epitopes. Here it's enough just to reproduce linear epitopes, which are easily reproducible for every approach, even bacterial one. So there is no need to create structural epitopes which are the exact 3D replica of the pathogen. Here is enough to recreate only the required part of the protein. If we talk about other infections, quite often to create on the bacterial to create recombinant genes, antigenes, sorry, on bacterial platform is very useful because they might, for example, in meningococc antigen that we are using, they are replicated exactly. There are no differences in the structure of antigen are observed. So here we have to admit that there are limitations for this type of expression, but here there are more advantages, more benefits, higher level of expression, cheap technology, high-speed technology. So the cultivation takes maybe 10 hours, yielding a large amount of biomass, enough for tens, hundreds, and thousands of doses. So here is we have a number of advantages which we believe outweigh the disadvantages. So we resolve it in most rational way, keeping all the technological advantages of this platform. Dear colleagues, if there are no further questions, as a moderator, I close this session. Congratulations to all the participants. Thank you for for interesting presentations and interesting questions, and we're looking forward to meet you again. Thank you. All the best.
Now we have the delivery or handover of the diplomas for the participation in this panel. Thank you very much. A strong applause for all of the panelists and all of us. We invite you now to pass on to the lunch hall in the lobby, and we will all be back as usual at 1:15 for the second panel, coordination between the national regulatory authorities and the private enterprises for health services and health products. Enjoy your meal.
solo la música escucho. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Prueba de sonido. 1, 2. 1, 2. Prueba de sonido. Sonido 2. Sonido 3. Ok, sí, sí, sí. No, estoy escuchando la música al mismo volumen. Ahí sí, bueno.
Ahí se bajó.
Sí, ahí se bajó. Ahí estoy escuchando. 1, 2, 3. Esta es una prueba de sonido, cabina English, Spanish. 1, 2, 3, prueba de sonido, español, inglés. 1, 2, hola, hola. Hola, hola, sí, sí. Sonido, 1, 2, sonido, ok. Ok, 1, 2, 3. Ok, ok, hola, 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 hola. 1, 2, 3. 1, 2, 3, 4, 5, 6. Probando sonido. 1, 2, 3, 4, 5, 6. Prueba de sonido. Sonido, ok, ok. Sonido 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Cabina número 1. Sí, ok. 1, 2, 3. Una prueba de sonido, sí, 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6, 7, 8, 9. Sound check, sound check. Sound check, 1, 2, 3, 4, 5, 6, 7, 8. 1, 2. Ya resolvimos la primera parte, estamos puenteando para acá, para que sea un sonido más limpio.
¿Cuál fue la primera parte?
Desconectar allá y poner el splitter. Hola, hola, 1, 2, 3. ¿Te está llegando bien?
¿El problema? ¿Problema de que necesitamos el piso?
1, 2, 3, 4. 1, 2, 3, 4, 5, 6. Sound check, sound check. Sound check, 1, 2, 3, 4, 5.
¿Habla?
1, 2, 3, prueba de sonido. 1, 2.
Hay que probar. ¿Habla?
1, 2, 3, 4, 5, 6. Prueba de sonido. Sound check.
El piso es el que nos está jodiendo. Ahí está bien. Está bien.
Sí, dos, 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 sí. Ok, sí, dos, sí. Y así.
2, 1, 2, 3, 4, 5, 6. Sí, sí, sí. 2, 2. No. 1, 2, 3, 4, 5, 6. 1, 2, 3.
1, 2, 1, 2, 1, 2. 1, 2, 3. ¿Y vos me escuchás ahí? Ok. Está bien. Si es así, está bien. Ahora, la otra cabina, le cambiaste también la entrada de audio. No, solo a esta. Está bien. Bárbaro. 3. Can you hear me? Testing English channel. Testing 1, 2, 3. Can you hear me? Testing English channel 1, 2, 3. Testing, testing, testing 1, 2, 3. English, English. Testing English 1, 2, 3. Testing. Hello, everyone. Please be seated so that we can start with the second session of panels. Testing English channel 1, 2, 3. Testing English channel 1, 2, 3. Now, welcome to the second panel discussion session, coordinating the RNAs and manufacturers. I'll give the word to Jose Vicente Coto, who is the International Advisor on Regulatory Matters. Thank you very much. Good afternoon. Before starting, I would like to thank you for the invitation. We've had a wonderful morning, but I have news for you because the best is still coming. So, we will start this panel's discussion in which we will have presentations. Some of the presentations will be done online, and then we will have a conversation, we'll have a debate. As we were saying, the second session is to cooperate in order to coordinate the national agencies and manufacturers. We also have two panelists that will join us from Russia. We also have the director of the SECMED agency from Cuba. We have the director of the Nicaraguan agency, and we also have the president of the Central American Chamber of Lab Manufacturers and Pharmaceuticals. They will be introduced briefly. First, we will get started with the first presentation. We have the honor today of having the presence of a representative of PAHO, who will be delivering the first presentation. She is Begonia Sagastui. Is headquartered or based in Washington. She's a specialist in public policies and international cooperation. She has over a decade of experience working for the Pan American Health Organization, and in recent years, she has been working with countries like Mexico, Nicaragua, Costa Rica, Trinidad and Tobago, and Barbados. Very recently, she was also working towards the regulatory development and legal development of some of the countries of the Caribbean. It is with great pleasure that I introduce Begonia to you. And she's also my comadre, and we are all ears. Begonia. It's a great pleasure to be with you, and it's also great to see many of my colleagues. I would like to greet Judith Rios, who was not able to join us during this session, but we also take this opportunity to thank you for considering us for this space because we value these opportunities a lot. I will share my screen, and if you could please confirm that you can see my screen. Perfect. Now I see I get the confirmation. Today we will talk about a topic that is central for the work, which is how we can strengthen regulatory systems, and because that affects in innovation and local manufacturing practices. This started 5 years ago with COVID, the pandemic, and this taught us a lot of lessons. As you remember, during the pandemic of COVID-19, it was evident the fragility of many health systems of our countries. There were significant interruptions in the system, and there was huge dependency on imports in order to access medications, supplies, and vaccines. We also saw a concentration of production of these supplies in only a few countries, which of course created inequities. And one of the examples of this was the vaccines. The main obstacle in order to provide vaccination was not the logistics, but the scarce level or the scarce amount of vaccines, and also the geopolitical tensions that we were able to see back then. And there were also some challenges with the supplies for the pandemic or during the pandemic. We also saw problems with the continuity of essential services. We can remember that the World Health Organization reported that one of every five countries was having interruptions for chronic disease treatments, and in 2020, 11 countries were at risk of being undersupplied. Just to give you a few examples, after the major or the worst part of the pandemic, many of the structural causes are still present, and we have new challenges that are compromising access. One of them is the reduction of official assistance for health development. In 2024, we reached the lowest point in the decade. Recently, in April 2025, WHO said that 1% the of these reported that there was a clear critical scarce levels, and the health services were being interrupted as a result. The pandemic showed that we don't have equitable access to medical technologies, and this the structural causes haven't been addressed. We want to talk about innovation in local production. In order to do so, we need to start from this reality, because we may think that the pandemic is way past us. Way past the pandemic, but we are still struggling with these issues. And the countries in the region have understood it. They want to overcome inequality. And the countries have mandated, as you can see on the screen, they have mandated the innovation and the strengthening of regional production of health technologies. The countries have understood that it's not only about producing, but also building productive systems where innovation, production, public purchasing, regional cooperation, and the final use of the project are all being coordinated. In this comprehensive approach will improve access to health technologies, but it will also be central to strengthen health and to promote the economic development, generate qualified jobs, and most importantly, also that is also important to make educated decisions by the countries to be able to say who are they going to produce for and when. There are also other elements that are key. We know that different countries in the region have consolidated production capacity for vaccines. We have regional mechanisms for cooperated purchases or joint purchases. There are also regional integrations that are very important for this. We also have a strong labor force as well. And we also have enabling factors, and there are challenges that we need to address. There's low investment in research and development. We are still depending on innovative medicines, and there are some barriers for technology in some of the countries. And this is a summary. So we need to strengthen, in order to strengthen the regional production, the countries of PAHO need to have identified the strategies to address these structural inequities. In order, we need to create an ecosystem where each component is important for the system itself, and we need to work in synergy. Within this ecosystem, the regulatory aspects are fundamental, they're instrumental.
Ahora recordemos cuál es su
So
rol
remember the
inicial
role
cuando
of the
llegó
regulatory
a
system
esta
within the
situación.
health
Los
systems in
países que
general.
han
We know that in
implementado
order
estas
to
políticas
reach universal health,
desarrollaron sus sistemas de salud
our health
pues
systems
con
need
productos
to have
médicos hechos en
medical
sus orígenes,
supplies that are
o sea,
available, that
asequibles,
are affordable,
disponibles en
and
todos
that
y
are
eficaces y
safe,
seguros. Y
efficient,
para
and
lograr
of
esto,
good
esta
quality. It
debería
is not enough to have
apoyar la
identified
implementación de
vendors
sistemas,
and funding
robustos sistemas
allocated. We
regulatorios.
also need a regulatory
Estos no
system that is
solo
based
buscan
on science
proteger a la
principles that can
población y
protect the
garantizar su
population and
salud y
ensure
transparencia.
technical, transparent decisions
Cuando
that
un
are
país, por
timely.
ejemplo, quiere
Also, if a
invertir o
country or region
desarrollar
wants to bet on innovation and production or scaling up existing
también
capacities,
tienen
they also
sistemas
need to have the
regulatorios
regulatory system in place because regulation should not be seen as a barrier or as control. It should be seen as a platform that enables and accelerates production to ultimately grant access to the population. What does it mean in reality? It means that we need regulatory systems that are efficient, that are technical, transparent, and timely along the life cycle of the technology. For instance, when it comes to development, they should offer early information and efficient processes to ensure clinical trials, even support during the registration, to have defined timelines for the production. And then the production should be inspected and validated uh, promptly, and there should be monitoring in the market to make decisions, quick decisions that are proportionate to any incident that may happen. Also, they should communicate very in a clear way with the producers, manufacturers, and the population. All of this is key for innovation because for states, this means a sovereign capacity to make decisions. For the population, it means trust, reliability, and safety. For the companies, it means a protection for their investments. So, this is why we say that regulation should be seen as something enabling, not something that's an obstacle. And there should be a bridge between the sustainable production and the population that will ultimately have access to these products. In the past decades, we've seen tremendous progress in technology in our region. Many countries have improved their regulatory frameworks, even countries that didn't have one. They created a regulatory system and they had even created an independent regulatory body and they have applied international standards, and there are still significant challenges. Here on the screen, we see, or we refer you to one of our publications, and we urge you to read it because we shared lessons learned about the most or the strongest regulatory organizations in our regions, and not only for the countries but also for the region. We see that there are funding challenges and there is a lack of mechanisms to recover the costs that it entails to regulate or to control, especially when you see the compare or when you compare it to the growth of the pharmaceutical market. Also, like in the case of pharmacovigilance, and also in some of the technologies, which is the case of the regulation of biologicals and medical devices. There are many opportunities as well to increase and improve transparency and cooperation of our regulatory systems in the region. And lastly, and very importantly, we see the need to have modern legal frameworks. And what we mean by that is that we should have legal frameworks that can balance between the technical capacity and the without forgetting that the ultimate goal is to ensure access to safe, efficient, and affordable products.
Y bueno, la OPS que participa o que hace en esto
What
para que
does
esto no
PAHO or
suceda
what's PAHO's
es
role
la
in it?
participación
We, the Pan American Health Organization, are the specialized agency from the Inter-American system, and we are the regional organization of WHO for the region. We have been working with the countries for many years, and especially in sanitary technology. For 2023, we created an innovation department for access of pharmaceuticals and technology, where we were trying to incorporate the three components, which is access, quality, and regulation, and innovation and production. So the creation of this department means that it doesn't mean that we didn't work on this before, but we wanted to do a comprehensive approach to promote, coordinate, and implement cooperation, technical cooperation work towards these technologies. Now, regarding regulation, we have different mandates coming from the countries. The most recent ones will be displayed here. We had a resolution in 2000 where the countries asked the organization to work on pharmaceutical regulation, which resulted in Red Farsa, which we will talk about later. Then in 2010, we had a resolution about the strengthening of regulatory entities of biological products and medications, and that had to do also with the role and the regulation in the Americas. And the most recent resolution from the countries was in 2022 in the Pan American Health Conference, which was a policy to strengthen the regulatory systems for medication and other sanitary regulations. So the first of these lines talks about promoting the creation of policies in order to strengthen governance and the governing of these factors. The next one had to do with the strengthening of regulatory systems to have consistent, transparent, and science-based processes. The third one is harmonization and convergence of the regulation. And the last one was the evaluation of regulatory systems based on world level tools created by WHO for the assessments. Here are some of the examples of the cooperation actions that we do from the networking work, as well as the technical resources and the support in the analysis and the creation of regulatory frameworks for regulation and some technologies, specific technologies that need certain specific support, like in the case of medical devices. I want to focus on two that are particularly important when we talk about innovation and production. Those are the work of harmonization and convergence and assessment or evaluation. For harmonization, we need to remember that one of the main challenges to make progress in production, we need to look at the existing regulation. As you know, each country has specific requirements and processes, regulatory frameworks, timelines in order to operate. But the pharmaceutical market is becoming more global, and it poses different challenges for the regulatory entities, like clearly those who are in the panel, when you need to make a decision about approving a product or not, especially a product that is registered in a different country, there may be difficulties of having access to technical data that was used to assess and make decisions about the product. And not necessarily we have formal cooperation or recognition and understanding of how that decision was made. This implies that many times we see countries that are starting from scratch, um, when even if it's the same product, and they use a lot of technical and human resources, and we miss opportunities for cooperation when these processes or requirements are too divergent. So it becomes even harder to develop production and share distribution strategies, and authorities face decisions. And of course, when we have limited resources, like in many countries in our regions, then this may compromise other actions that are critical for the countries, like pharmacovigilance, and other regulatory actions. So here is where harmonization and convergence for the regulatory bodies becomes very important. What we mean is that for regulators to take international best practices and cooperation by respecting the sovereignty of each state and their regulatory bodies. And we need to understand that there may be legal requirements that are different from one country to another, but they could go aligned progressively in order to facilitate the dialogue and the cooperation. And without a doubt, this is key for production because when countries move towards harmonization and convergence, they strengthen their collective capacities for oversee manufacturing processes, and that makes the registration and vigilance more efficient because then you can expedite in the processes and you create stable conditions, coherent conditions for local quality production, which can ultimately be exported to the region. So it makes a lot of sense to promote harmonization and convergence, especially when we have the idea of creating productive systems that really meet the national needs. This is a space for the Red PARF, or the PARF network is a space for, it's an initiative that is led by regulatory agencies with the support of PAHO. It's a space where we create consensus and we promote specific projects to work towards harmonization and convergence, following a peer cooperation scheme. It's a unique opportunity to get better prepared, more cooperative regulatory bodies. We need to strengthen the network in order to adapt to the new realities and the new needs of our regulatory bodies.
A second important initiative I would like to stress has to do with the uh, maturity of regulatory authorities, especially those who are regional reference. As I mentioned before, this concept was formalized by state members of PAHO in 2006 in 2010, and what is sought here is to create a framework with a common evaluation agreed to by the countries to identify the different levels of maturity that already exist and in which authorities can share and show their technical capacity as to become reliable reference. So it seeks to promote these convergence, trust, facilitate the use of the different networks and so on, so as to avoid overlapping. The experience, as you can see on the screen, its history, the different years, and this is now being updated. And today, the RNR is adapting to the most recent standards and updating. And now we are working with the regional evaluation according to GBT, and the regional reference authorities can be key to promote the regional production. They're now internationally recognized. Its strength of our authorities is recognized internationally, and that makes it a lot easier as regards the way forward, and it's fundamental to continue to support this framework that has been so unique in the Americas. No other part or region of the world has anything similar, and so it really is important for both renovation and production. And to wrap up, I would just like to say we've really advanced a lot in the past 2 decades, but the challenges are still there. And we need to work together on this. We are consolidating the progress made. We're accelerating the processes of harmonization to ensure that no country in the Americas is left behind. That means there has to be strong, sustained technical cooperation, political will, because we're talking about regulations, but we cannot forget that the world of strategic alliances that participate is absolutely critical, and a space such as this one, producers, association of patients, and in general the forging of strategic alliances is very important. We as PAHO invite you to join this effort. I wish to reaffirm our commitment to continue working with you, because it's only with strong regulatory and reliable systems can we advance toward a region with much more innovation, more production, more access for everyone. We thank you very, very much for this space to present you the work done by PAHO on these matters, and invite you to please continue with these dialogues so we can continue with the mandates that we have received from our state members. Thank you kindly. Thank you very much, Begonia, for your excellent presentation. We will now continue in the program, and the next person that will address us is Vadim Merkulov. And while that is prepared, I just wanted to make a couple of comments here. The first is that there is a code so you can gain access to, if you have any questions for the panelists, and you have it off there to the side so that you can ask questions, and the microphones will be open too for those who want to say it personally, they may. And secondly, I would like to thank the virtual presence of those who are accompanying us in distance. We have reports that there are people that have been connected since the morning, so we greet all of those who are listening to us from abroad. So Vadim Merkulov, Deputy Director of the Center for the Scientific Investigation of Medical Use Products from the Ministry of Health in Russia.
I would like you to help us because only Russian language can be heard. The Spanish speakers cannot hear Spanish. Only the Russian audio is audible, so we cannot hear the translation. The sound is interfering here. The Russian sound is interfering, and we cannot hear the interpretation in Spanish or any other language. Uno, dos, tres. Prueba de sonido en español. Maybe. Prueba de sonido. ¿Escuchan mi español? Prueba de sonido. ¿Escuchan mi español? Are you going to sit?
A ver, sí, voy a intentar. Perdón. Sí, ¿y en qué canal en español lo hago, no? Sí, sí, ajá, ya. Sí, confírmeme que puede hablar. Sí, desgraciadamente hemos tenido problemas de sonido. ¿Se me escucha ahora? Se me escucha perfectamente. A ver. Sí. Коллеги, наладили перевод, если что, можно попробовать еще раз. Dígale, por favor, que podemos ir. Sí, dígale. Sí se puede. Ahora bien, parece que funciona. Vamos a intentarlo una vez más. Díganme, por favor, si se me escucha. Sí, le escuchamos perfectamente. Párenme si pasa algo. Tengo que empezar desde el principio, ¿no? Dígale a Cristina, por favor, para que ella interprete al ruso, porque de aquí no puedo hacer al ruso. Sí, siga, por favor, doctor Merkulov. ¿Empiezo desde el inicio o tengo que continuar? No, continúe, por favor, continúe. Es que prácticamente he dicho nada, o vuelvo a dar la presentación. No le escucho. Sí, claro está. Estimados colegas, estimados participantes del Congreso, tomado en consideración la gran distancia que nos separa, yo entiendo perfectamente que no ha habido la posibilidad de escuchar toda mi ponencia debido a los problemas técnicos, problemas de traducción, problemas de sonido. Entonces, como me piden los organizadores del Congreso, voy a resumir mi ponencia y voy a explicarles una vez más que para promover los fármacos de alta tecnología modernos a nivel nacional y a nivel internacional, hoy en día es imprescindible ah, desarrollar a los reguladores, que tiene un rol importantísimo, ya que tiene que ir acompañando el desarrollo de cada nuevo producto de alta tecnología desde el momento de su obtención in vitro hasta su comercialización en el mercado o en los mercados de nuestros estados.
Toward that end,
Para este fin se
we
crean
create different
distintos
mechanisms,
mecanismos que tienen como su
various mechanisms
objetivo acelerar la implementación de esas
to
tareas.
facilitate those
Así, por
issues.
ejemplo, en la
Here in
Federación
the
de
Russian
Rusia, en
Federation,
los últimos
we
años
recently
hemos
have
tenido una
witnessed a
serie
number of
de
changes,
cambios
and as
y
of today,
nosotros hoy en día
we are
realizamos el acompañamiento del desarrollo de
following the
estos
development
fármacos
of
importantes para
such
el sector
important
de salud.
uh,
Para cumplir con este objetivo
for
hemos creado
healthcare
unos
medical
grupos de trabajo o los paneles
medications
que se dedican
in
a
specific
trabajar
concrete
las
areas.
líneas de desarrollo
For
concretas.
example,
Para ponerles
We
un
have
ejemplo,
a
tenemos un
task
grupo de
force
trabajo que se dedica
for the
al
development
diseño
and
y al
registration
registro
of
de
medications
fármacos
based
basados en
cell-based
células,
cell-based
fármacos celulares.
medications. These
Estos
medications
fármacos se
are
utilizan
used for
para el
treatment of
tratamiento de las enfermedades oncológicas de la
of
sangre,
blood
antes
cancer,
que nada en los
first of
niños.
all among children.
Se trata de
These
T
are
linfocitos
modified T
modificados
lymphocytes
y LINCAR-T
and CAR
tecnologías.
technologies.
Y estos mismos fármacos,
To the
este
same
grupo
group of
incluye también los
drugs
fármacos
belong
para la
medications for
medicina
regenerative medicine,
regenerativa. En particular,
namely
la que
the
contribuye a la
restoration
recuperación de los
of
cartílagos de las
the
articulaciones.
joint tissue.
En el marco de estos grupos de
Within the framework of
trabajo
these
que
groups,
están formados
including
por
leading
peritos
experts,
líder, por
leading researchers,
investigadores líder, por los
developers and
desarrolladores y los
manufacturers.
fabricantes de los fármacos, se
So within these
desarrolla
groups,
la
they
discusión de
are
los
discussing
problemas más
the
relevantes
acute
en lo que
issues
se refiere al
related to the
registro de los
registration
fármacos
of
de
these
este tipo.
medications. Another similar
Otro
group
grupo de este tipo es el grupo
is
de
dedicated to the
desarrollo de
development
las
of vaccines
vacunas a base de
based on
ARNM.
RNA.
Se trata de
These
las
vaccines, as of today,
vacunas que hoy en día tienen una importancia muy alta. Es un
are in the
producto de
front line
vanguardia que ayuda a la
and are
terapia de
extremely
muchas
important for the therapy
enfermedades
of
oncológicas.
a number
Un
of diseases,
ejemplo de ello
of
es el
oncological
cáncer
diseases.
colorectal
For
del
example,
colon y
the
del
most vivid
recto.
example is
Se
colorectal
trata
cancer.
de dos
These two
grupos
groups
que en la Federación de
in the
Rusia
Russian
están
Federation are
presididos
headed by
por
me,
mí
these
y
two task forces.
yo veo como
And
algo muy
it is of
interesante
interest
el poder
to be able
intercambiar
to
con
share
ustedes mi
our
experiencia
experience with you, with
y
our colleagues,
también
and probably
es muy
your
interesante que
leading
sus
researchers in the area
investigadores
and your
líderes en este
experts
ámbito, que sus
may
peritas formen
join
parte de
our
nuestros
task
grupos
force
de trabajo
in order to
para
discuss
poder discutir los
the
asuntos más
most
palpitantes relacionados
acute
con estos
issues
fármacos
related
de
to these drugs.
vanguardia.
So,
Y
briefly,
también en mi
I have described
ponencia
in my presentation
he dedicado
the
un
express
periodo
mode
breve
of
al
registration
registro de
of
los fármacos que son de la
medications
mayor
which are
importancia
of
para el
extreme
Sistema
importance
Nacional
for
de Salud.
national health care. These registrations
Este registro se realiza a
are
base de datos
performed
clínicos
with
incompletos
incomplete
con
clinical
la
data
completación
with a
posterior de
subsequent
los estudios
completion
clínicos
after
después de
conditional
este
registration.
registro previo.
As of
Hoy en
today,
día en la
in the
Federación
Russian
de
Federation,
Rusia,
such
estas
novelties
innovaciones han permitido con
allow
la máxima
to launch
eficiencia
these
hacer salir esos
medications
fármacos a los
to the
mercados
national market with the
nacionales.
highest efficacy.
Basta con
Enough is
decir
to say
que
that
en el último año la
in
Federación
the
de
last year alone, in the
Rusia ha
Russian Federation, we have
registrado
registered
más de
over
450
450
fármacos
new
nuevos.
medications.
Con ello
This
voy
brings my
terminando mi
presentation to an
ponencia
end. I would like
y
to wish
quiero
all of you
desearles a todos ustedes, a todos los participantes, ponencias interesantes en el resto del
reports
Congreso
and
y
presentations
también
till the end of
desearles
the
a
Congress,
todos ustedes
and
la
wish
salud
all of
y la
you
paz.
health and peace.
Gracias a todos
Goodbye,
ustedes.
dear
Hasta
colleagues,
luego.
and
Me
goodbye,
despido
my
de
friends.
ustedes, estimados colegas y queridos amigos. Gracias.
We will now hear from Elena Dimisova. She is the head of the Division for the Production of Medicines at the Ministry of Industry and Commerce. I would ask Elena to give a brief introduction, basically for everyone's knowledge, how the regulatory structure functions in the Russian Federation. Please, Madame. Could somebody please put up the presentation? had problems with the audio, and now we don't see the screen, but hopefully this will be solved forthwith.
Then, while they are working on the slides, on the image, on the display, I will probably skip, I will probably pass on my turn. So we may listen to the next report, maybe. Thank you very much. No, we should not have any issues with the sound, probably with the images neither. Since I do not understand Spanish, I will proceed in Russian.
¿Ahora se escucha? ¿Se escucha? Sí.
Then I will try briefly to
Bueno,
tell you what we are doing
entonces
in the Ministry
voy a
of Trade and
hablarles
Industry
sobre
in
la
the
estructura
area of the
regulatoria en
regulation
Rusia, la
of
regulación
medications.
de los fármacos.
First of all, I would like to emphasize the
Primero ante
fact
todo
that in the
quisiera
Russian
decir que en
Federation
la Federación de
there
Rusia
are a
tenemos
number
varias
of
agencias,
agencies
varias
involved in
instituciones
the
que se
registration and
dedican a la
circulation
circulación de
of drugs
fármacos,
and
como por
medications.
ejemplo el Ministerio de
For
Salud,
example, Health
que se
Ministry,
dedica
they
a la
are the leaders
pericia
in
de los
the
fármacos. En el marco de este trabajo también
investigation
expedimos las
of the
autorizaciones para
medications,
estudios
permissions
clínicos,
for clinical
evaluamos la
research,
documentación,
etc.
etc.
Ministry of Trade and
Ministerio
Industry, which
de
I have an honor of representing here. We are involved in production licensing. We control the GMP rules and license requirements. As has been mentioned in the first presentation, it's definitely very good when we have some established rules of regulatory approaches and market launch. So for 10 years already, the Russian Federation and the four more countries, the Republic of Belarus, Republic of Armenia, Kyrgyz Republic, and Republic of Kazakhstan, were united in Eurasian Economic Union, setting shared rules for the circulation of medications. The main issues of this common market are presented in the agreement on the general principles, which has been signed in Moscow in 2023. On December 23rd, 2014. We have also developed a number of requirements for the production. They have the unified rules for pharmaceutical inspections have been determined, and for the quality of work of pharmaceutical inspectorates. There are other decisions for this common market, including decision number 77, which described the expertise of the manufacturing. We also have another regulations, including the laws on the state control, as well as decrees which are describing the implementation of certain legislative norms and legislation issues. For example, if you look at the law on the main types of activity, you will see that the licensing of the production of medications is mandatory. However, Decree 686 describes the specific requirements for the company applying for license. License is mandated not just by the requirements of the Russian Federation, it's also a requirement of the Eurasian Economic Union, uniting 5 countries. It also in accordance with international practice as well as Federal Law 61, which is the basis for the circulation of medications in Russia. So both in the international legislation and in the Russian legislation, the compliance with the GMP is mandatory. So, according to the law of licensing of certain activities, the requirements are those mandatory rules and requirements which have been established by the governmental decrees based on the requirements of the legislation of the Russian Federation and articles of international agreements. So licensing there are requirements for licensing, which I presented here. So it has to be a vested person who is part of the staff of the company, who verifies that every medication launched into the market and circulating in the market matches the requirements, and the GMP regulations have been met during the production. Also, the manufacturing site has to be included in the register according to the uniform rules of the Eurasian Union, covering all 5 countries. Also, it mandates the number of number of specialists with the required educational levels, and the licenses issued by the government of the Russian Federation, they have no term limit, but that does not mean that the manufacturer, once receiving a license, may rest assured for the rest of his life, because once he obtained the license, he is subjected to the legislation of state control. The state control, so obtaining the license is the easiest probably for the manufacturer, but then if the manufacturer wants to manufacture some new drugs which are not mentioned in the license, so it has to make changes, amendments, and also usually has to confirm the confirmation to the requirements within 2 years. You used to have scheduled inspections. Then after pandemics, the inspections have been banned, but the new legislation changes have introduced the new requirement, which requires the manufacturer to confirm compliance every year. Once the manufacturer fails for whatever reason, he didn't submit the application for the inspection or failed the inspection, but even if he did not submit the application for such an inspection, then his license is suspended and his GMP certificate is suspended. So without GMP certificate, he is unable to proceed with his activity, with his production. So it's definitely his responsibility.
The periodical confirmation that I have mentioned before, and the quality the licensing procedure and the certification procedure. What does that mean? Even if a manufacturer has in its license or certificate the right to produce, manufacture certain product, then the Ministry of Healthcare can initiate and can conduct such an inspection before the registration procedure is over. A little bit of explanation. I would like to tell you what our inspectors are guided by when they perform the inspections. Two main decisions for EAEU, ruling number 83, the rules and the order of conducting an inspection, and number 82, which purpose is for each inspector to be guided by uniform single rules and approaches. After the national decisions were made, the ruling by the government was issued, and the Ministry of Industry and Trade was the one in charge of putting those decisions into practice. And then, based on this document, an order was issued on the pharmaceutical inspections procedures and the list that includes all the inspectors that can perform such audits. The structure of it looks like this. It's quite complicated, but I will try now to explain to you. The Ministry of Industry and Trade is the main agency that has specific department that is in charge of conducting such inspections. And inside of this department, we have all the pharmaceutical inspectorate or pharmaceutical inspectors, and we also have institution of GMP and pharmaceuticals, and it has its own department in charge of conducting inspections, and they have their own list of inspectors. They perform all the duties related to the inspection of licenses, all the respective documents. It's about GMPs, pharmaceutical licenses, certificates, and all the other documents. That is the job of our inspectors to ensure the proper circulation on the market. The responsibilities include the conduction of inspections, and the institute is responsible to also perform the inspections, sometimes together with the inspectors from the Ministry of Industry and Trade. That is how this scheme works. And here we see the scheme for receiving a license. How do manufacturers obtain it? And many times we have mentioned COVID pandemic today, and it was a shock for everybody, but we can say that it has had some advantages, and we made progress in terms of digitalization. For Russian manufacturers, they can submit all the documents on the portal that is called Gosuslugi, or government services, state services. Because in the past, it used to be like that. A manufacturer from Vladivostok had to fly to Moscow to submit all the documents, and it was rather difficult. But now they can just open their computers and submit all the documents in an electronic form. For manufacturers that are based outside the Russian Federation, so far we do not offer this service. They have to to submit the documents in paper form, or maybe they can send it by post as well. In order to get GMP certificate, they need to provide a list of pharmaceutical products that are being manufactured or that are planned to be manufactured. The pharmaceuticals that have been already developed, the production cycle has been validated, and these are the products that are ready to be launched on the market, that is completely ready for registration and clinical trials. And it is obligatory to provide these documents for all their production facilities. There is a guideline for the submission in accordance with the decision, the procedures. It's not a very large document, but it describes the activities of the enterprise, all the flow charts, the logistic chains, etc. Here on the map, we wanted to show to you that since 2014, since GMP requirement became obligatory, the number of the inspections that have been conducted, the overall number over the last 10 years, roughly, more than 3.5 thousand inspections for Russian manufacturers. And as for the manufacturing facilities outside the Russian Federation, we started performing those inspections, conducting them in 2016, and the figures are quite comparable there. And we try to encompass the maximum number of manufacturing facilities outside Russia. And now in Russia, we have 542 enterprises located in 73 regions of Russia. That is my brief review of what we do, activities that we do, and I will be glad to answer any questions you may have.
Muchas gracias, Elena. Al escuchar
thank Carla for the effort that you are making here in Nicaragua, and I encourage you to continue. Carla Estela Perez is with us as well. She is a attorney at law. She has worked in the pharmaceutical and has worked in El Salvador and is now the president of the Federation of the Central American Pharmaceutical Industry. So we will have a balance of 4 presentations, and I will put them on the table an issue and ask them to interact, although Elena is welcome if you would like to comment on anything that is to be discussed. So the first matter I'm going to put on the table here for discussion is what should the main interest be that guides the relationship between regulatory agency and the manufacturers, because this is sought today so that the authorities are able to strengthen their collaboration and then see what the nature of the relationship between regulatory agencies and manufacturers should be. So from how it's seen from the point of view of the industry, how it's seen from the point of view of the regulatory agency. So Olga, if you would like to start. Thank you, Dr. Koto. It's an honor for us to be here and to participate at this 9th Congress. It's the 3rd time that SECMED has been represented at this Congress, and we feel very much at home. We think we're really part of this great family, the family of our Russian colleagues and our Nicaraguan friends as well. So thank you very much for the opportunity. We feel that to talk about cooperation between national regulatory authorities and manufacturers is to talk about public health as a shared purpose. And this goes along with what Dr. Koto was saying. No, we all have a common good, and that is to work to put at the disposal of our people or medicines and or medications, and of course to give them access. In the presentation we heard from Dr. Begonia, we were very pleased to hear the overview that she gave us regarding the strengthening of a regulatory system and the role that is played in an effort to achieve that these medications really get to the population, but with the quality that is also necessary. Also important is the relationship between manufacturers and the regulating agency, and that's what I've been asked to comment here at this panel. So in the case, for example, of SECMED, we would like to share with you the experience that we have had, especially during COVID, when in the initial phases and the early stages of research and develop of these candidates for vaccines, and we had very strong interaction with the manufacturers of these vaccines. And this interaction between regulators and regulees helped us to shorten the different time periods between the different stages, preclinical, research, development, and so on, all these stages, because we worked very closely together and we accompanied the process in such a way that they provided information to us with the different information packages, and in that way the process was accelerated. I think the pandemic was what really forced us to work in this way. Never before had we had an interaction as strong as at that point between the manufacturer and the governments or regulating agencies, and that was due to the pandemic. Since then, we have established a process that is now habitual, and we anything new, anything that's innovative, the new technologies and so on, as they are developed and as the regulator then, when they receive a dossier, already knows the information, they've been kept up to date on what is being done, so you don't have to have long evaluation processes. And we think, right here we have a best possible example of coordinated work, very interactive work between agencies, regulating agencies, and manufacturers. We have, for example, the St. Petersburg Institute, Mechnikov, and the regulating agency in Nicaragua and Cuba, and they were working together. It's here it is in this room, in this very room, and this progress has been achieved thanks to this effective communication between all that began a long way back, but that we are keeping up to date. We're updating it, we're strengthening it every day more, and it has been really, very advantageous to have this exchange, and has made it possible for these vaccines to reach this happy ending where they're already pre-qualified by the World Health Organization. And this just goes beyond borders, because these pre-qualified vaccines will be available for the entire regions of the Americas. And I think that that is something that we feel proud to be for this result that is now already available for the people. No, so the Menshnikov anti-grip or anti-influenza, with excellent results. And we also, the strengthening of the regulating agency has a direct impact on the production capacity. And when a regulatory authority advances to greater levels of maturity according to the World Health Organization, the evaluation of authorities by the GBT, they become a strategic actor for the health and economic development of a country. So we cannot stop. To the contrary, we must continue and to force the regulatory agencies to heighten and improve their standards even more, so that these manufacturers are able to put their products on the market, but with the trust and reliability that is necessary. The relationship between the industry and the regulatory agencies must be based on constant dialogue, transparency, trust. We need regulatory frameworks that, without losing their rigor, are agile and adaptive, able to respond to innovation, to the health challenges that we face. From SEGMED, we reiterate our commitment with the construction of collaborative spaces between regulators and manufacturers, in which we together promote solutions that strengthen our local and regional capacities. To cooperate to promote health is not just a motto, it's an urgent need and a shared responsibility. Thank you kindly. Thank you, Olga. When we think of Cuba, we think of a regulatory referent, no? I mean, obviously, there's been work among Rafael and other people that have really helped us to appreciate this process that Cuba has followed, but they really are a referent for us, and we're being accompanied also by countries, especially from South America and Central America, and Cuba, of course, working very closely with Nicaragua. And in fact, some of the technical operational problems that have been found in Nicaragua have then been solved by the presence of SegMed in some areas where it was necessary to get this technical support. So I give the floor to Carla, who is the director for the Department of Pharmacy at the National Sanitary Regulation Authority of the Ministry of Health in Nicaragua. Good afternoon to all. Allow me to thank you for the invitation to tell us about our experience as the National Regulatory Authority for Nicaragua. And of course, we congratulate those who have organized this event because, as Olga was just saying, we have been strengthening our sanitary regulation and making a collaborative effort together with the Mechnikov Institute to be able to achieve what we did, which was the pre-qualification of the anti-influenza vaccine. So obviously, basically, this could not have been done without transparency. There needs to be trust, active participation between the two entities, the two sides, and as a regulating entity, the process to strengthen us as a regulatory authority. So based on this experience, which goes back quite a few years, we now in Nicaragua, our regulating authority is committed to working and review, structure, organize its normative framework and develop new capacities, new regulatory processes. We've been working on this, and we feel very proud that we have been able to achieve what we have indeed achieved thus far. We have acquired training with the support of Russia, the support of Cuba, of PAHO, and it has helped us then to strengthen our staff members. And working then together with the laboratory, we have heard what the problems are that they have, and we as authority, what the decisions are that they need to take, and how we can help them, how they can help us, and how we can work together, and because it implies the strengthening of all regulations. So as a regulating entity, we commit ourselves to continue in the process of transparency, convergence, of drawing together, working closely with manufacturers, with the principals, with all the different stakeholders to ensure that the product that is commercialized and especially that is exported from Nicaragua is up to standard as regards quality. Thank you very much, Doña Carla.
It is truly interesting to see the evolution process in Nicaragua. In the case of the authorities in Nicaragua, Nicaragua is one of the countries with an autonomous agency, as administratively as well as technically, and has been able to separate from specific individual interests of different state agencies, and this positions the Nicaraguan authority in the management of this area in which mechanisms are looked for and found for better management. We can also speak of a transformation in the industry. In 15, 20 years, there have been changes in this area, and now there is a common interest between the authorities and the pharmaceutical industry, because both are interested in offering access to medication to the population, and this is a common goal. And thus, we also want to listen to Carmen and what she thinks about this relation. Good afternoon. Thank you very much. It's a pleasure to be here. First of all, I would like to thank for the invitation to the Mechnikov Institute. I hope I pronounced the name correctly, for opening this and organizing this important forum about biotechnology and vaccines. which is an important topic for all of us. And as you have mentioned, the regulating agencies as well as the regulated actors are walking in the same direction. And you have asked for the main goal and purpose that we both should be looking for. And I think that we from the industry are interested in access to medication that is safe and of high quality. You remember that we have lived through the pandemic and we consider that we have overcome it, but many lessons have been learned from it. One lesson was that due to the closure of the borders, the only medication and drugs that we could have access to were locally produced drugs in each of our countries and in Central America. Therefore, the strengthening of the pharmaceutical industry and the local production of drugs is not unimportant. It is not irrelevant. It is, quite the contrary, a strategic ally for crisis, not only to overcome a crisis, but to prevent a crisis through our constant and continuous work in our health systems. And our purpose and our work, our joint work, should be focused on that aim. Dr. Olga already mentioned our joint work and that it is necessary to have an open and honest dialogue in which we share whatever each party can contribute to this aim. And then we have to look for a form of cooperation in joint measures that helps us to advance, both of us. The health authorities, Dr. Cotto, when he came to the National Department for Medication in 2012, he took us by the hand and pushed us a little bit in order for the pharmaceutical industry to strengthen their capacities. As the regulatory authorities become more autonomous in their actions and decisions, then we could advance and make progress together. At that point in time, the National Direction of Medication, that is now changed the name to a super department, and it changed the course of things. And it started a new form of working, much more executive, much expedite, much more direct, cleaner, and without many complications, which allowed the industry, gave it a chance to have the interest in making the investments it has made. From 2014, the pharmaceutical industry in El Salvador has invested more than $100 million in infrastructure, in equipment, and in trainings. And this is not all of it. They are continuing with their investments and their reconversions, as well as the growth of quality standards that are ever higher in the industry. And the industry of the entire region has done so in Honduras, Guatemala, Nicaragua, and Costa Rica as well, in order to be able to apply the good manufacturing practices, norms, and standards in their own work and investments. But this was a result of an open dialogue, of open and honest commitments about work that can be undertaken and investments that will be made. And the regulating agencies, as well as the regulated actors, were able to advance on that basis. Thank you very much, Carmen. I would like to bring a second topic on the table. Exactly now, in this interaction between the industry and the regulatory agencies, there's one issue that is not less important. In order to strengthen the national regulatory system, it is crucial to advance towards a harmonization of the regulatory functions at the regional level. In this context, how can we describe the relation between the regulatory agencies of each country, and what concrete steps have to be made to align and approach the functions of these regulatory agencies between the nations. Now, this is not only of interest for the regulating agencies, but also for industries, because many of these industries work with different governments, so they are in touch with different agencies, and it's a kind of a headache to have to comply with the requirements of all these different agencies. And we would like to be illustrated by Olga. Now, we all know that many years ago we have started with harmonization, and we know that harmonization is a very complex process, and now we are walking towards more confidence in the regulating agencies. This is a highly important element, that the regulating agencies should be able to enforce these mechanisms, because we are clear that there are inequities or differences between the different agencies. In moments of crisis, we have been able to see very clearly that products had to be evaluated, and that there were pending products that had not been approved yet, and in the agencies there weren't enough human resources to accelerate this process. So we worked on the basis of confidence towards other regulatory agencies, which allowed us to put into practice what our goal, which was the access, and still is the access, of safe and high quality drugs. So if one agency has already evaluated a product, then another agency can ask for the information, can ask for summaries of evaluations of these products, and on the basis of this information, then take a decision on the basis of this confidence and trust towards the other agency to have a more expedited accreditation and approval process in the other country. Of course, we always will need all the information, a complete documentation, but we are talking about the evaluation process. This can also be applied to the inspection processes. Why should it be necessary to travel to other countries and waste money and time if there are already agencies in those countries that are recognized authorities, and we can find mechanisms between these regulatory agencies in order to exchange information and develop procedures that will give our decisions a solid fundament in the information and decisions of other agencies. Now, this work, we have made this kind of work, very close work, very solid work with Russia, with the GMP Center. We have signed memoranda of understanding, and we have defined that we are in a process of exchange of information about our regulatory frameworks, and this is what we want to get at. We want to reach this recognition of the inspection framework, inspection reports that the agency in Russia is doing, in order for us not to have to send our own inspector over there to make his or her own inspection. This is what we think is a good idea, because we all need to be able to do this in the Central American region. There are many agencies in Latin America that recognize the reference authorities. However, this has cost a lot of work in order for the reference agencies to recognize each other at their own level. This is a reality today. Other agencies recognize us as the SECMED, and there are others as well. And we have to continue to work in this direction to strengthen the confidence and trust between the regulating agencies. Thank you very much, Olga, and I would like you to deepen a little more on this topic that Olga has mentioned. Last year, no regulating forum omitted bringing up this topic for each for an agency that has a very small budget. The trust and reference they can um, take from the work of other agencies is, of course, very valuable. Please, can you say something about this? Yes, generally, on the basis of what Dr. Olga just presented, the national health regulating agencies have decided that they needed to work on this basis for the approval for new drugs and vaccines. So we developed an administrative resolution which expressed the recognition for reliance on other agencies. And as Dr. Koto said, in reference to all of our agencies, we need to simplify the approval processes. We need to help and to facilitate to the evaluator the review of the documents that had been presented in order for the response and the answer and the decision to be much faster. Apart from the proceedings, the confidence in other agencies helps us not to avoid having to travel to other countries and do our own inspection. So it helps us to be able to trust the evaluations and the documents and information that comes from other regulating agencies. Now we started with the drugs. Now we are working on some medical equipment and devices, and in the future we are going to cover other services and products as well. Now, before I pass the floor to Carmen and Elena, I would like to recognize that among the participants we have the director of the Honduran Health Regulatory Agency. You're welcome, Doctor, and I would like to hear from you what you think about reliance. How does the Honduran authority think about this? What would you like to say about this? Just very shortly. Thank you. I am sorry for taking you by surprise, but I am quite sure that it will not be difficult for you to answer here. Thank you very much. Thank you for the invitation. It's a pleasure to be here with you, with well-known colleagues and friends. Yes, this topic is very important, and the understanding that we have to have from the regulatory point of view, as Carmen has already said, in conjunction, in joint work with the industry, and now talking about the reliance and the confidence that we are using as regulating agencies. I think this is, I know that this is a strategy that has helped us to develop our capacities and strengthen the capacities of our human resources in order to be able to grow in this area, such as the GBT area that is so important. And so we are able to recognize the hard work that has been done by other regulatory agencies. And as Dr. Carla has said, this helps us to give us access to medication and to other products that are of vital importance for the Nicaraguan population, the local population. So this technical cooperation has been a cross-cutting axis, and we are still in the process of developing it. We have many initiatives that we are working on with industries in different countries. We are also working with different agencies in other countries in the same field. And this is a process that is ongoing. And we are trying to find a regulatory convergence at all levels. Thank you very much. Please go ahead, Carmen. It's my turn, right? The work in Central America has been marked by Central American technical regulations and all the coordinations and cooperations that have been made in the framework of this convergence. It is not only about creating new regulations, but also the criteria for the interpretation of the norms, because we sometimes may have the exact same law, but we apply it differently. And we were just talking about this with the doctor from Honduras. Even inside one and the same institution, different members or officials apply the regulations differently and the criteria as well. So it's not only towards the outside world, it's also inside for the institutions. And I think the most important thing is to work closely, be in close contact. Reliance is very important, although it's not all, and not, but there are some things we can delegate to others, and this will strengthen other topics that are more critical. And we can solve things immediately that are critical, and delegate other things or postpone other things that are not so critical. In the case of El Salvador, the sanitary authority is in the process of being certified at the 4th level, and the industry is also the industry will have more trust in our work, because we will be able to comply better with the higher standards, because the industry always looks for higher certifications, and this will favor the industry because it will open more markets to it and give it access to other countries and populations, and thus obtain better prices for our products. I thank you, Carmen. I will ask you for a little patience, because we will now give you some time for questions. We had some delays because of technical problems, and now we would like to have a summary by Elena about what, or a response by Elena about what
Thank you.
we have been
Thank you
talking
very
about.
much. So we've touched upon many topics, the procedure that is based on trust principles, the mutual recognition, the harmonization inside the region. And I can tell you a little bit more about harmonization of legislation inside the region, those 5 countries that I've mentioned, and it was shown on my presentation. And as for this market, since this document was adopted, 10 years have passed, and for a long period of time, each country has had its own way, its own principles, legislation, approaches, and we are facing a lot of difficulties in terms of interpretation, the speed of decision making, but we don't give up, because we have not only our institute, but also GMP Academy, where we train specialists and experts. A lot has been said today about the need for the dialogue between the regulator and the industry, and I completely agree with that. I firmly believe that a licensing authority, as a licensing authority, we work in order to prevent counterfeit products entering the market. As Carmen has mentioned, many experts interpret norms and rules in various different ways. And it is not only about the experts interpreting it, but also the industry, even inside an enterprise. During an inspection, for example, we find some incompliances, and we ask them, the representatives, the employees, whether they have understood that, and they say that yes, we did understand, but then we ask for the detailed explanation of how they understood that. And there are different types of interpretation. And last year, we launched a cycle of education. Every 3 months, we train experts from various enterprises. We choose the most problematic topics for us as regulators. Now, for enterprises, for example, some incompliances that we see during our inspections, and we try to provide this information in a good way for all these people to understand it properly. That is our experience.
Thank you very much, Elena. Thank you. Applauses, please. Applauses to all the panelists.
We still have some minutes left. If there is any further question or contribution by the audience, please go ahead. Yes, there is a question here from Eddie Lopez Guido of the National Security Systems. 5 basic steps to advance towards level 3. And that's a question for Olga. 5 steps to advance towards the 3rd level. If Olga does not know the answer, then no one of us will know. Well, let's see. The evaluation process to become a reference authority has some indicators and sub-indicators. In the region of the Americas, there's a process that has been ongoing for several years now, where many of the agencies have passed through this process and have been able to reach level 4. I would not say that there are only 5 steps. I think that they are many small steps that have to be taken to become a reference authority. Now, today, we are working on the GBT. GBT is a very rigorous process. It's a process of the WHO, and this process is very complex, has 9 modules with many indicators and sub-indicators, and in order to become an authority listed by the WHO, this will cost us a lot of work, but we are on the way. SECMEC is preparing itself, and at the end of this year, we will apply for it. But if you ask me about 5 steps, I would like to say that there is a fundamental step, which is the human resources. The agencies need very well educated and trained, competent staff with lots of experience, and in all the basic functions. We need to be able to show and demonstrate that we are able to comply with and execute all our basic functions. We need a solid regulatory framework, and we need to be able to demonstrate this. And most of all, we have to have laboratories for quality control, for analytical trials, and if we don't have our own laboratories, we need to be to have some laboratories that we can contract for these purposes. But it's not only about having an installation or facility. The human resources are, from my point of view and experience, a fundamental factor. So, well, if you want to know more about other further elements, then we could have some email exchange for that purpose. Thank you very much. And if you allow me, I would like to add the following, that apart from the human resources development and training, for us, it is also a critical factor because there is no history of training for regulatory agencies, but we have mechanisms to channel all this knowledge and the factors to improve the processes, and also the legal structure is very important, the legal basis, in order to be able to know where the basic regulation is going to walk. So for several years, we have been working on this in Nicaragua. Is that correct? Right. Any other question from the people here in this hall? Anybody wants to take the floor? Very well then. I would like to thank you. It has been an extraordinary event, and also the participation has been good. The quality of the panelists, of course, has been excellent. And I hope that you have enjoyed this day as much as we have. And we'll see each other tomorrow again. Thank you very much. Now we will hand over the certificates to our panelists. We say goodbye now for today with a strong applause. Thank you very much. We thank you for your participation in the first day of the 9th International Pharmaceutical Congress, Russia-Nicaragua 2025. And we invite you for tomorrow. We start from 8:30 to 9 tomorrow morning with the inscription of the participants. And we ask you please to be here punctually so we can start 9 o'clock short and have our first session until 11 o'clock in the morning. And I wish you a good afternoon and many blessings. Goodbye.